Christina, and this video is on cell cycle phases. You're at home lounging on the couch, enjoying your post-exam half day. What do? Catch up on your favorite medical TV drama, of course. The doctors are treating a 35-year-old female patient with multiple cancers. She has breast cancer, renal carcinoma, and a past history of leukemia. Her father has a history of lymphoma and had osteosarcoma as a child. Her younger sister also has adrenal cancer. You shout at the TV, Li-Fraumeni syndrome, has to be. Now, what tumor suppressor gene is affected here, and at what phase of a cell cycle? We're going to be talking about the different cell cycle phases. In the next few slides, you should be able to 1. Identify the different cell cycle phases and functions. 2. Recognize the regulatory processes in place. And 3. Describe the clinical significance cell cycle inhibition may have. So what are the different cell cycles? Well, if we go back to 7th grade biology, we can recall the most general phases of the cell cycle are interphase and mitosis, where interphase is the growth of a cell and mitosis is where cellular division occurs. Interphase consists of gap 1 or G1, which is the first growth phase following mitosis. S phase is a synthesis phase where DNA replication occurs, and gap 2 or G2 is the last growth phase before mitosis begins. Mitosis, or M phase, consists of mitosis itself, which is where cellular division occurs, and cytokinesis. It is the shortest phase of the cell cycle. Mitosis has a few different components that I'm sure you all recall. To quickly summarize, you have prophase, prometaphase, metaphase, anaphase, and telophase, which I remember by double P MAT. Now, it's very unlikely you're going to get a question on your board exam that specifically asks about these individual steps, and it's better to cohesively understand what happens in mitosis and cytokinesis versus just memorizing these specific steps. Upon entering M phase, cellular growth stops at this point and all the focus is on cellular division. Mitosis represents chromosome separation and cytokinesis represents separation of all cellular components. At the end of this phase, mother cells are divided into 2 daughter cells, genetically identical to each other and to their parent cell. A phase not really part of interphase or mitosis is the G0 phase or gap 0 phase. This is also known as a quiescent phase of the cell and represents the resting phase, when a cell is neither growing nor dividing and has simply left the cell cycle. Permanent cells such as neurons, skeletal and cardiac muscle cells, as well as RBCs remain in this phase indefinitely and regenerate from stem cells. Stable or quiescent cells such as hepatocytes, lymphocytes, and periosteal cells are also in G0 phase, but when activated can move on to the G1 phase. Cells that divide very rapidly, such as bone marrow, gut epithelium, skin, hair follicles, and germ cells, all collectively known as labile cells, do not enter the G0 phase. Instead, they enter G1 and rapidly divide and progress down the cell cycle phases. So can cells rapidly grow and then divide without going unchecked? No, right? Or our body would be completely out of whack and we'd have tumors and cancers growing like rapid fire. There are different checkpoints in place that control transitions between different phases of the cell cycle. So if something is wrong and this cell should not be dividing, it will stomp on the brakes. One family of proteins that regulate the cell cycle are cyclins and cyclin-dependent kinases, CDK for short. Cyclin-dependent kinases are always around in all phases of the cell cycle, but sometimes are active and sometimes are not. What activates them? Well, as the name suggests, cyclins, which is why they're called cyclin-dependent kinases. There are several different cyclins present at different phases of the cell cycle. Cyclins will activate specific CDKs, forming cyclin CDK complexes. These complexes then activate other proteins via phosphorylation and allow for cell cycle progression. For example, in S phase, they'll activate proteins to initiate DNA replication. The G1 phase is variable and can last indefinitely, but once cells transition to the S phase, they're committed to division. Therefore, the most important transition to regulate is a G1 to S phase transition. This is where tumor suppressor genes such as retinoblastoma or RB and P53 come into play. They work to inhibit or suppress this transition. So what would be a reason a cell would want to halt cell cycle progression? Well, one of the most important reasons is DNA damage. P53 is one of the most notorious genes for detecting and responding to DNA damage, which has earned its nickname, the guardian of the genome. If P53 notices something's wrong with the DNA, it will prevent the cell from dividing, thus preventing any tumors from occurring. When DNA damage is detected, P53 will stop the cell cycle and initiate DNA repair pathways to try to fix the damage. If the cell is able to repair the damage, the cell cycle can restart. However, if it can't, P53 will initiate apoptosis, because it's better for the cell to die than to risk developing cancer. Retinoblastoma, or RB, also prevents a transition from G1 to S phase and works with P53 to do so. Basically, if P53 senses DNA damage, it induces P21. P21 is another inhibitory protein, part of the CIP-KIP family, which stands for CDK interacting protein kinase inhibitory protein. P21 then activates RB through hypophosphorylation, and RB works to halt progression from G1 to S phase. Only when it's inactivated by adding more phosphate groups will it allow the cell cycle to continue. And guess what type of protein adds phosphate groups to RB in order to allow the cell cycle to continue? Our friend, the cyclin-dependent kinases. Tumor suppressor genes like P53 and RB follow the two-hit rule, meaning that both copies of the tumor suppressor gene have to be mutated in order for significant neoplastic transformation to occur. This is because if only one copy is mutated, the other one is generally sufficient enough to regulate the cell cycle and prevent tumor growth. However, P53 is so powerful that more than half of all cancers have a loss of function mutation in P53. Remember the disease from the introduction, Li-Fraumeni syndrome? This is an autosomal dominant disease that results in multiple cancers starting at a very early age. You tend to see the classical SBLA cancer syndrome, which stands for sarcoma, breast, leukemia, and adrenal gland carcinoma. But you can also see lymphomas, renal carcinomas, as well as brain tumors. Alright, let's do a quick flash quiz. The tumor suppressor gene P53 and RB prevent the transition from the blank phase of a cell cycle to the blank phase. These block the G1 to S phase transition because remember, once you get past that step in the cell cycle, it's pretty much committed to go through the rest of the cycle. Alright, so the bottom line. The cell cycle consists of interphase and M phase, where interphase includes G1, S, and G2 phases, and M phase includes mitosis and cytokinesis. There are several regulatory proteins in place that serve as checkpoints. These include cyclins, CDKs, as well as tumor suppressors, P53, and RB. And lastly, the most important phase of the cell cycle to regulate is the G1 to S phase transition, and mutations in this regulatory step can result in cancer formation, for example, Li-Fraumeni syndrome. That's all for cell cycle phases. Thanks for watching. If you enjoyed the video, don't forget to give a thumbs up.